So let me move to the uh, sixth chapter. So in the morning we discussed about first about classification, wild relatives, centers of origin, then about plant classification and diversity of plants. Then we move to the morphology of roots, stems, leaves, flowers. Then flowers can pollinate. Next, after pollination, seed production. So let's look at seed, germination, dormancy, and dispersal. This is the intended learning outcomes. Describe the paths of monocot and dicot seeds. Explain hypogynous and epigynous germination. Identify factors affect seed germination. Then seed dormancy and factors affect seed dormancy. Then methods that can be used to break dormancy. Then explain the importance of tree dispersal and methods of dispersal. So these are the ILOs. Uh, what is a seed? Seed is a mature ovule. So when pollen transferred from anther to the stigma of a flower, it will develop a pollen tube, and finally the nucleus will enter the ovule. So seed is a mature ovule and it has an embryo. Ultimately, that fertilized ovule will develop into a ovule. Ovule will develop into an embryo. This is the result of sexual fertilization. Then what is germination? Germination is the resumption of the growth and development of a seed. So if you look at the seed, it's given in the book. These are the different parts of a seed, right? Outermost cover, then the point at which the seed attached to the stalk is called helium. Then there's a small pore called micropyle. It helps to absorb water. Then inside the seed, there's a radicle which grows into the root system. Then plumule, which grow into the shoot system. Then certain leaves have large endosperm, stored food. Then all the leaves have, seeds have an embryo. This is the embryo, right? Those are the parts. During seed germination, what will happen? Radicle will develop a root. So once the root system is developed, the seed will anchored into the soil so that this radicle anchored into the soil so it can absorb water. So what will happen? When water is available, metabolic activities is activated within the seed. Stored food will be break into simple compounds, right? So when the favorable conditions are present, seed will produce radical, then radical can absorb water, or the helium can uh, absorb water. Then once the water is absorbed, seed will swell and it induce the formation of hormone called gibberellin. With that, metabolism starts. So the starch, stored food like starch, can become simple, less complex food. So the root system and shoot system can develop, right? To begin with, initially the seed will not have any root system, but so seed can absorb water. That will initiate metabolic activities within the seed, right? Not the radical before seed form the radical. Seed can absorb water through the process called imbibition. නිපාණය මගින් ඇටයකට ජලය උරා ගන්න පුළුවන්. This activate formation of gibberellins. Then gibberellin will produce enzymes so that stored food will break down into simple compounds. So the radical will develop downward, plumule will develop upward and produce roots and shoot system. So it's a simple process. So dicot leaves have two cotyledons, you can see. Upper part is called epicotyle. Above part to the cotyledons are called epicotyle. Hypo means below. Hypocotyle means below part of the cotyledons. Then seed germination are of two types, epigeal germination and hypogeal germination. Epigeal germination means api Brahma Parohane. Here, cotyledons raise above the soil. Cotyledons raise above the soil. So we call epigeal germination. Opposite is hypogeal germination. Hypo means below. Cotyledons remains under soil, below soil. So it is called hypogeal germination. Ado Brahma Prarohane. Right? This is details are given in the book, right? What are the factors that affect seed germination? We can put factors into two groups, internal factors and external factors or environmental factors. If you look at the external factors, like four factors can be identified: temperature, water, oxygen, then light or darkness. Depending on the seed type, certain seeds need dark period, certain seeds need light. So those are the four factors that affect seed germination. Those are the external factors. So initially seed will absorb water through a process called imbibition. Nipane magin jala uga gannawa. Then it trigger gibberellin or hormone activation, then enzyme activation, then seeds can germinate. Then oxygen, temperature, for each and every seed, there's an optimum temperature level, right? It's usually minimum 10 to 12, maximum 40 to 42, optimum is 30 to 37. Room temperature, just above the room temperature is usually best. Then light or darkness, depending on the seed type. Then what are the internal factors responsible for seed germination? Seed viability. What is viability? Ability of a seed to germinate when we provide optimum conditions. Then what is longevity? Longevity is the period within which a seed show renewal of growth or germination. There is a limit period or longevity. Seed dormancy is the one that usually seeds are unable to germinate in a specific time period under combination of environmental and maybe internal factors. Until the seed get correct conditions, seed will not germinate. That is due to the dormancy. Otherwise, when the mango seed is attached to the fruit, it can germinate, but it is not going to happen. Right? Seed, we are going to look at dormancy in detail in a minute. Then germination rate is the percentage seeds germinated out of the total. You called it germination rate. In seed packets, it is indicated. Right here, you can see usually rice seeds are dormant. If it is not dormant, what will happen? It will start germinating while attached to the mother plant. So we will not be able to consume it. So each and every seed has some sort of dormancy mechanism so that it will survive until the favorable conditions reach. Because in the paddy plant, when the before harvesting, conditions are very dry. Season is dry. Water is not available. So they will never germinate under these conditions. It will take some time to uh, initiate germination, right? So it is good for us. We can consume it. So dormancy is a mechanism to prevent germination. බීජ සුප්තතාවය කියන්නේ බීජ ප්‍රරෝහණයෙන් නවත්තන්න තියෙන මෙකැනිසම් එකක් during the unfavorable conditions when the probability of seed survival is low. So usually when a plant produce fruits and seeds, sometimes conditions are unfavorable. The condition may be very dry. So if seeds start germinating, what will happen? It will not survive without any water. So there's a dormancy period where seeds will not germinate. Why dormancy is important? To prevent pre-harvest sprouting. Before harvest, otherwise seeds can sprout. Then also act as a survival mechanism in plants. Overcome unfavorable environmental conditions. Both are very much similar. Allow time for seed to dispersal, because though a plant produce seeds, immediately seed will not disperse. So as an example, in the case of mango, maybe bats should visit the plant and they should grab the mango seed, mango fruit. They will fly away long distance and they will eat and drop it, so that it needs some times, right? So allow time for dispersal. So this is pre-harvest germination of rice. This is an issue, right? So it is not good. So dormancy is one way good. Dormancy can be classified into endogenous dormancy, that is inside embryo dormancy, and exogenous dormancy caused by conditions outside the embryo. Endogenous dormancy, exogenous dormancy. Right, now let's look at exogenous dormancy, outside conditions which affect germination. Physical dormancy, it can be put into three groups: physical dormancy, mechanical dormancy, chemical dormancy. What is physical dormancy? It is due to the impermeable seed coat. Sometimes seed coat may be very thick. It may not allow water to penetrate in, so seed cannot germinate. Mechanical dormancy, sometimes seed coat may be too hard for the embryo to expand. So seed coat is very hard, so that embryo cannot come out to initiate germination. Chemical means there could be some chemical compounds present in the covering around the embryo. We should remove these things before the seeds trigger germination. We can soak seeds in water so that those chemicals can be washed away from the uh, seed coat. Then the seed can initiate germination, right? Those are the three different germination uh, exogenous dormancy types. What are the endogenous dormancy? Due to the factors related to embryo. Physiological dormancy. This prevents embryo growth and seed germination. So, due to physiological dormancy, embryo growth is prevented. Hence, seed germination will be hindered. So, once a chemical change occur, then it can overcome. Then, morphological dormancy, embryo may be underdeveloped or undifferentiated. So, you will not be able to rectify it because embryo is not developed. Combined dormancy mean combination of both morphological and physiological. So those are the three categories of endogenous dormancy. So we discussed three categories of exogenous dormancy, outside factors to the embryo, and three factors within the embryo: physiological, morphological, and combined. So next, let's look at how to break the seed dormancy. අපි බීජ සුප්තතාවය අයින් කරන්නේ කොහොමද? There are few methods. First one is you know that there are different dormancy related to seed coat. Seed coat may be very hard or impermeable. So we can soften seed coat and other covering. We called it scarification. Scarification කියලා කියන්නේ seed coat එක ගොඩක් හයිය වෙන්න පුළුවන්. අපි ඒක කොහොමද මේ බුරුල් කරන්නේ? ජලයට පාරගම්‍ය කරන්නේ නැත්නම් එම්බ්‍රියෝ එකට එළියට එන්න පුළුවන් විදිහට හදන්නේ කියන එක. There are different methods, mechanical methods. First, mechanical scarification. We can mechanically rupture the seed coat by using a knife or by using sandpaper. We can mechanically damage the seed coat, as you can see here. You can scratch it, you can use a sandpaper so that you can damage the seed coat so water can penetrate in and it can trigger germination. That is called mechanical scarification. Scarification is softening seed coat, right? So first one is mechanical. Next one, hot water scarification. We use hot water, like depending on the seed, we can use different temperature for different duration so that it will uh, damage to the seed coat and it will allow water penetration by hot water, or we can use acid. Then we call acid scarification, like sulfuric, nitric acid can be used. You can soak seeds in acid, or warm moist scarification, that is keeping seeds in moist, warm soil or unsterilized sand for several months so that seed coat may become softened due to the microbial activity. It can decompose, then the seed can absorb water and it can germinate. So these are the different scarification method, that is softening seed coat methods: mechanical, hot water, acid, warm scarification, right? Uh, Next, stratification. Previous one is called scarification. This is stratification. What is stratification? It is the process of treating seeds. Seed treatment. Why? To stimulate natural conditions. For what? Then seeds will experience conditions similar to the outdoor environment, favorable conditions. Stratification is a process of treating seeds. Why? To stimulate natural conditions. So therefore, seeds must experience, so these conditions must be experienced by seeds before they germinate, right? There's a small mistakes in the words. Then refrigerated, so stratification can be, or the stimulation can be given in different methods. Just like scarification, you can keep seeds in the refrigerator, so you call refrigerated stratification. Then outdoor planting, when you plant seed outdoor area, it will stimulate germination. Then leaching, that is washing seed with fresh water for a few minutes or few hours, depending on the seed. Then pre-chilling, store water absorbed or imbibed seeds at 5 to 10 degrees centigrade for 5 to 1 week. Pre-drying, subject dried seeds to 37 to 40 degrees temperature treatment to 5 to 7 days prior to germination. You can expose with light. You can apply acid. You can apply hormone like gibberellin. So those are stratification methods. In other words, stimulation methods. So seed dispersal. Why should seed dispersed away from the mother plant? When a seed dropped under the mother plant, what will happen? Competition will be very high. So seeds will not germinate. So usually vector will carry seeds away from the mother plant. ගහක් යට බීජ වැටුනොත් පැලවෙන්න පුළුවන්. පැලවෙලා පැල 1000ක් විතර ඇති වෙන්න පුළුවන්. එකක්වත් සර්වයිව් වෙන්නේ නැහැ. මොකද කම්පිටිෂන් වැඩි, වතුර නැති වෙන්න පුළුවන්, පොහොර නැති වෙන්න පුළුවන්, ලයිට් මදි වෙන්න පුළුවන්, right? So transporting propagules is very important, and it can be happen through biological or abiological vectors. So usually seed survival is higher away from the parent plant because there could be competition with the mother plant. So usually seeds have different mechanism to move away from the mother plant. You can see here large number of seedlings under the tree. Imagine the number. None will survive after 1 year. They all will probably die because of the high competition. Therefore, seeds evolve strategies for dispersing away from the mother plant. What are those strategies? It could be its own mechanism or internal mechanism or external mechanisms. If you look at these seeds, they are just like insects. So birds can come and take these seeds and they can move away. And some seeds like cashew, they have fruit. There is, it is, and this kotamba, it has fruits, so that stored food. So animal like to eat. First one is gravity. You know, due to the gravitational force, fruits fall away from the mother plant. So sometimes uh, later it can washed away with water or animal can disperse. First one is gravity. Second, explosive mechanism like rubber or impatiens kudalo, it can explode. So seeds move away from the mother plant. Then some seeds have wings, so seed can be carried away from the mother plant, far away distance, like horror, Dipterocarpus. Water can be used as a media to disperse seeds. Then animals like wild animals like wild boar or deers in certain plants, they have hooks, so they can attach to the animals so that it can move away from the mother plant. Then humans sometimes carry seeds knowingly or unknowingly. Unknowingly, like it can attach to the shoes, like with soil, so we can move long distance with cars, with ships. Seeds can carry away with human clothes, right? So those are the different methods of uh, seed dispersal. Right. With that, let me move to the last two chapters: plant cell, tissues, and structure. ILO, at the end of the lecture, you should be able to describe prokaryotic and eukaryotic cell, the difference between prokaryotic and eukaryotic cells. ප්‍රාග්ඥාස්ටික සහ න්‍යෂ්ටි ජීවීන් අතර වෙනස කියන්න පුළුවන් වෙන්න ඕනේ. Components of the plant cell and its functions. සෛලයේ කොටස් සහ ඒවල කාර්යභාරය. Different types of cells and functions. You know, all the cells are not similar. They are of different types. They modified to perform different functions. Then types of tissues and functions. Then structure of the leaves. Then the structure of monocot and dicot root. So this, of course, if you are interested, you can read the book, but I am not going to ask this section during the exam. Structure of monocot and dicot stems and root. This part I'm not going to ask in the exam. So for the exam, you can skip it. But if you like, you can read it. This is a cell. If you look at the prokaryotic cell, Pragnyastica cilia, these are the genetic material, inherent material. They are not enveloped. They are freely present in the within the cell, like bacteria, blue-green algae. But if you look at higher animal plants, all the genetic materials are enclosed with membrane-bound nuclei. So this genetic material is enclosed in eukaryotic cells. So eukaryotic cells have a true nuclei. This is a comparison. Eukaryotic usually both are multicellular. Here eukaryotic nucleus present, in the other one absent. Membrane-bound organelles present, like mitochondria. All are covered with the membrane, double membrane. Then nuclear membrane is selectively permeable, but in prokaryotic no nuclear membrane. Cell wall present in plants and fungi. Here vacuoles present, both vacuoles present. Cell division is by mitosis. This is by binary fission. Like in bacteria, they always divide into two, dvigunanaya. Sexual reproduction present and by meiosis. But here no sexual reproduction, only transfer of DNA. Right? Those are the differences between prokaryotic and eukaryotic cell. So you should be able to draw and label parts of a cell, right? You should know it by heart. Um, so, right, you should be able to draw a cell like this, right? So in a cell, this is called middle lamella, red color. Then inner to that, you have a cell wall. Inner to that, you have a cell wall, right? Interior to cell wall, you have plasma membrane. This solution is called cytoplasm. Then cells have a nucleus. Within the nucleus, you may have nuclei, one or few nuclei. Then, so the nucleus sometimes it's difficult to draw. This is called endoplasmic reticulum. These structures are fixed to the nuclei. There could be several nucleus, right? Then there could be chloroplast. Then there could be mitochondria. There could have a large vacuole. Then small lysosomes. Then from this one, sometimes Golgi bodies can be produced. Golgi bodies, right? So this is a typical plant cell, right? It's difficult to draw, but try to learn how to draw a cell and to label all the parts. It's very important. But if you look at a cell has living part and non-living part. Cell wall is non-living. Protoplast is living. What do you call protoplast? Protoplast consists with plasma membrane. Inner to the cell wall, you have a membrane called plasma membrane. Then the solution. Inside the solution, we call cytoplasm. Apart from that, in the cytoplasm, you get a lot of organelles like mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, like things. Then large vacuole and a nucleus. All these things are called protoplast. So this part is living. Cell wall is dead, non-living. You have to remember it. What are the living parts of the cell? Cell wall is non-living. Protoplast is living. It consists with the outermost membrane we call plasma membrane. Then inner is filled with the fluid we call cytoplasm. And the organelles like mitochondria, chloroplast are embedded in that one. Then large vacuole, then nucleus. Cell wall. Uh, if you look at the cell wall, two cells are joined together with the middle lamella. මධ්‍ය සූස්තරයෙන් සෛල දෙකක් එකට සිමෙන්ති වෙලා තියෙන්නේ. බදාම් වගේ සයි වෙලා තියෙන්නේ. So this is the middle lamina, red color middle lamina. So using middle lamina, those two cells are glued together. It consists with calcium and magnesium pectate. Then inner to that, you have a primary cell wall. First produced cell wall is primary cell wall. Composition, you should know the composition. It is given here in the book as well. Mainly hemicellulose, cellulose, pectin, protein, and polysaccharides. In some plants, there is a layer between primary cell wall and plasma membrane. You call it secondary cell wall. So when a plant form a primary cell wall like this, And there could be, this is the, let me draw in the enlarged version. This is the plasma membrane. This is the primary cell wall. So another layer can be formed in between primary cell wall and secondary cell wall, right? Another layer can be formed between primary cell wall and secondary cell wall, right? Primary cell wall and secondary cell wall. This is, sorry, primary cell wall and plasma membrane. This green color one between primary cell wall and plasma membrane. That is called secondary cell wall. It provides rigidity or strength to the cell composed with cellulose and later deposited with lignin and suberin. So it becomes very hard. So if you look at cells, this is one cell. So innermost plasma membrane, in between primary cell wall, outer wall and plasma membrane, you have secondary cell wall. And two cells can be connected with, we call plasmodesmata. ප්ලාස්ම බන්ධ මගින් ශරීර දෙකක් සම්බන්ධ වෙලා තියෙන්න පුළුවන්. එතකොට නිකුත් ද්‍රව්‍ය හුවමාරු වෙන්න පුළුවන්. Right? Material can be exchanged through plasmodesmata. Here the binding agent, cement between two cells. This is cell number 1, cell number 2. Both are glued together with middle lamella, madhya sustra. What are the functions of the cell wall? It gives a shape, protect the cell, communication, barrier to undesirable large molecules. Protoplast. So the protoplast is a thing which include plasma membrane, cytoplasm, nucleus, and vacuole. We discussed it earlier. Plasma membrane is, or cell membrane is, semi-permeable. So it acts as a semi-permeable barrier. වර්ණ පාරගම්‍යතාවයක් තියෙනවා ප්ලාස්මා පටලයට. ඒතකොට සමහර දේවල් ඇතුළට එන්න පුළුවන්, සමහර දේවල් එළියට යන්න පුළුවන්. තෝරාගෙන ඇතුළට එළියට යවීම සිදු කරනවා. So they act as a permeability barrier so that certain things can come in, certain things can move out. And it helps in for the cytoskeleton or to give support for the cell. Then cytoplasm is a fluid inside the cell. It is filled with different organelles like mitochondria, chloroplast, right, endoplasmic reticulum. They are embedded in this cytoplasm. This is cytoplasm, right? This is mitochondria, chloroplast, endoplasmic reticulum. This is the nucleus. So this is a picture of a cell. And what are endoplasmic reticulum, ER? It linked to the nucleus and it goes like this. It has large number of membranes and sometimes it can move away out and connect with the next cell as well. There are two endoplasmic reticulum, smooth ER and rough ER. Smooth ER, no ribosomes. Rough ER, ribosomes present. In rough ER, it can synthesize proteins. In smooth ER, it can synthesize lipids. Apart from that, they can transport metabolites to neighboring cells because this can extend from one cell to the other cell through plasmodesmata. Then we have plastids. Plastids are pigments which gives color. First one, chromoplast. They are responsible for giving color other than green color. Like if, when leaves become senescence, it turns yellow color. Fruit ripening, green to yellow to red color. Petals are blue color, red color, yellow color, orange color. That is due to the presence of chromoplast. So sometimes carotenoid pigments can be present and it can give different colors. As I mentioned, yellow, orange, red color. So this chromoplast can be developed from the chloroplast. As an example here, at the younger stage of a fruit, there could be chloroplast. So it's green in color food. When it's become mature, chloroplast can convert into chromoplast. So it will give a different color like orange or yellow, whatever it is. So this color is due to the chromoplast. Next one is chloroplast. I'm not going to give details of chloroplast because Professor Abhay Singh has covered it under crop physiology, right? So it is present in leaves. They are responsible for the green color. And they are help in photosynthesis. I'm not going to discuss the content. It's given in the book and Professor Abhay Singh has already covered. Then leucoplast. Third category is chromoplast, chloroplast, and leucoplast. Chromoplast give different colors other than green. Chloroplast gives green color and photosynthesis is the major function. Then leucoplast are colorless pigments. They store food. If it store carbohydrate, starch, we call amyloplast. As an example in potato. When they store proteins, we call it alluroplast. Then mitochondria, once again, Professor Abhay Singh has covered it, right? So it's a double membrane organism. So it helps in the respiration process. Next, endoplasmic Golgi bodies. Golgi bodies, this is the Golgi bodies. They produce vesicles so they can deliver certain compounds from one place to the other place. Site of deliveries. So function, modifying, storing and packing proteins, transport lipids, creation of lysosomes, manufacture of biological polymers containing enzymes. So these are very active organelles in plant cell, Golgi bodies or Golgi complex. Ribosomes, hope you can remember, they were located in the endoplasmic reticulums and they can synthesize protein. Then come the nucleus, the main organism, organelle. It is covered with the membrane in eukaryotic plant. It inside include genetic material. So they can transfer from one characters from one plant to the other through this genetic material. This nuclear membrane can be fixed to an endoplasmic reticulum. So it can extend and finally to a neighboring cell. So chromosomes are present. Chromosomes are the material responsible to carry genetic information from one cell to the other cell. Then all the plants have a larger vacuole. It covers about 90%. And there's a sap here we call cell sap, covered by a membrane we call tonoplast. So it's store water, salt, toxic material, etc. Usually plant cell have a large vacuole. Then lysosomes. Lysosomes are formed by Golgi complexes. They have membranes, sets of membranes containing enzymes. So enzymes can digest unwanted material. So they can uh, digest unwanted large molecules. Okay, that is all about cells and different parts of a cell. Then similar cells together form tissues. and they might perform specific function. Let's look at different types of tissues. Parenchyma, right? So, sorry, let's look at the cells, different types of cells, right? Uh, once a cell is formed, it can specialize to perform various functions. සෛලයක් හැදුනට පස්සේ ඒක විශේෂණය වෙන්න පුළුවන් එක එක කාර්ය ඉටු කරන්න. දැන් අපි බලමු මේ විවිධ සෛල වර්ග මොනවද කියලා. Let's look at different cell types. Parenchyma cells, මුර්දුස්තර සෛල, they are live, they have large vacuole, oval, round or elongated shape. Function is photosynthesis, storage and secretion. So parenchyma cells are very active. They produce food, storage function, secretion function. Collenchyma cells. If you look at the collenchyma cell walls are thickened. Here you can see the cell and the corners are thickened. These cells contain chloroplasts, so it can photosynthesize. Apart from that, it can provide mechanical support. Sclerenchyma cells, all the cell walls are thickened, so it will provide mechanical support. So parenchyma cell wall is not deposited with any material. Collenchyma, only corners of the cell walls are deposited. Sclerenchyma, all the parts are deposited with lignin. So function is mechanical support. They are not active, physiologically active. මුද්දුස්තර සෛල, ස්ථූලකෝණාස්තර සෛල සහ දුඩස්තර සෛල. මුද්දුස්තර සෛලවල බිත්තිය තැම්පත් වෙලා නැහැ මොනවත්. ලිග්නින් කොලෙන්කයිම මුද්දුස්තර ස්ථූලකෝණාස්තර සෛලවල කෙළවරවල් වල බිත්තිය තැම්පත් වෙලා තියෙනවා. ඒතකොට ශක්තිය දෙන එකත් එකකින් කරනවා. දුඩස්තර සෛලවල ජීවයක් නැහැ. ගොඩක් වෙලාවට එකකින් ශක්තිය ලබා දෙන එක තමයි කරන්නේ. Then there is another type of cells called flanking stomata, we call guard cells. Here you can see guard cells. It regulates the transpiration. Water loss is regulated by this. This can either close, as you can see here, or open. Then epidermal cells, outermost layer of the plant body is epidermal cell. It can produce trichomes, hairs, or just cells. Then vessel elements, that is xylem and phloem elements. If you look at xylem element, they are of two types, xylem vessel and tracheids. They transport water and giving strength. These are the xylem vessel. Then sieve tubes. Phloem is made out of sieve tube cells. These are the cells. Their function is water transport. Xylem cells, transport of water and mineral plus giving strength. Sieve tubes, phloems, transport nutrients. So these are the different types of tissues, then different types of cells. So group of cells of common origin and common function is called tissue. එකම තැනකින් සෑදුන ඔරිජින් එක සමාන නම් කරන කාර්ය සමාන නම් ඒ වගේ සෛලයට අපි පටකයක් කියලා කියනවා. So there could be different tissues, meristematic tissues and permanent tissues. Permanent tissues means they do some permanent work. Meristematic tissues means they are always dividing. විභාජක පටක සහ ස්ථිර පටක කියලා පටක දෙකක් තියෙනවා. විභාජක පටකවල වැඩි නිතරම සෛල නිපදවීම. Let's look at the permanent tissues. So in permanent tissues, all the cells are similar in structure and function, like epidermal tissues, parenchyma tissues, collenchyma tissues. Those are a group of similar cells we call tissues. Like xylem tissue, phloem tissue, those are group of similar tissues. Group of similar cells from tissues. Next group is meristematic tissue. It means it is always dividing. So it is produced at the shoot tip, root tip, in the stem, etc. Right? as you can see here, for the stem apical stem elongation, root elongation, then to increase the width and length of the tissue, these meristematic tissues are important. If you look at the meristematic tissues, if it is located at the apex of the root or shoots, we call apical meristem, agrastha vibhajaka. If it is located inside the stem, it will help to increase the length. So it's called intercalary meristematic tissues. Then it could be parallel and lateral. So it helps to increase the diameter, width of the plant. They are called lateral meristematic tissues, right? So meristematic tissue could be apical, intercalary, or lateral. So when you look at tissues, we can identify three basic types of tissues. Dermal tissue, ground tissue, and vascular tissue. Outermost cover is called dermal tissue. If you look at the stem, outermost cover, dermal tissue. Innermost is vascular tissue. It could be consist with xylem and phloem. In between, we call ground cover, ground tissue. What are the function of dermal tissues? It form the outer skin of the plant body. So it protect the plant cells, it prevent water loss, and it can come comprised of hairs, trichomes, etc. So protection is the main function. Then ground tissue, it consists mainly with parenchyma cells. So storage, you can remember the function. So photosynthesis, secretion, etc. are the function. Then vascular tissues, that is xylem and phloem. Innermost, we discussed about xylem and phloem. Right. With that, those are the different types of tissues. Now let's look at the structure of leaves. Outermost cover of the leaf is epidermal cells, as we discussed, right? It is cover the internal part. Then this could have a waxy cuticle, then stomata, guard cell, mesophyll cells, palisade mesophyll, spongy mesophyll. Let's look at the LS of a leaf. I told you outermost layer is upper epidermis. Lowermost layer is the lower epidermis. අපි චර්මය උඩු අපි චර්මය සහ යට අපේ චර්මය it could be covered with the cuticle. Then you have a palisade layer, palisade mesophyll layer. It has chloroplast, so it can photosynthesize. Then you have spongy mesophyll cells. Those have spaces in between. These plants also, these cells also can do photosynthesis. Then there are vascular bundles, and lower side you have stomata. This is the line diagram. Remember, you should be able to draw and label all the parts. This is the upper epidermis, udapicharmya. It can be covered with the cuticle layer. Then palisade layer, it may contain chloroplast, so green in color. Then spongy mesophyll cells layer. In between, you have spaces, air spaces. Intercellular spaces, then lower epidermis. And in the lower epidermis, you may get stomata. Its structure, that is the structure of leaf, structure and growth of dicot and monocot stem. I told you that I'm not going to, I don't expect you to know it for the exam. If you like, you can refer it, right? So I'm going to skip it. Structure of roots and shoots. If you are interested, you can read it. Right. With that, let me move to the last chapter, cellular reproduction. At the end of the lecture, you should be able to describe the cell cycle and its different stages, then you should be able to describe different stages in the mitosis and meiosis, anunana vibhajana and unana vibhajana, then phenomenon of crossover, avataranaya, then describe the importance of mitosis and meiosis. අනුනන සහ උනන විභාජනයන්ගේ වැදගත්කම කුමක්ද? Cell cycle. So when a cell produces daughter cells, it can divide and produce another set of daughter cells. This occurs as a cycle. Cycle of growth and dividing. Cells reproduce by how? By cycle of growing and dividing. But do we call it cell cycle? So it's a cyclic process. First, it has three stages, three main stages. First stage is called interphase. During interphase, cell grow, cell produce more DNA, cell will produce more mitochondria, chloroplast, all the material necessary for cell division will be produced at this stage. Cell growth stage or interphase. Then after that, this will move to the mitotic phase, or we call nuclear division phase. During that time, duplicated DNA will divide, nucleus will divide and produce two cells. It will undergo another 4 stages: prophase, metaphase, anaphase, and telophase. Then once the nuclear division is over, the cell will enter cytoplasmic division stage. We call cytokinesis. Divide වෙනකොට stages 3ක් කරා යන්නේ. Interphase නැත්නම් සෛල වර්ධනය වන අගවදියේ මෙතෙන්දී DNA සහ සෛලේ තියෙන ක්ලෝරෝප්ලාස්ට් මයිටොකොන්ඩ්‍රියා ගුණණය වෙනවා. ඊට පස්සේ න්‍යෂ්ටිය බෙදෙනවා කොටස් 4ක් කරා. ඊට පස්සේ සෛලෝප්ලාස්මේ බෙදෙනවා. Right, this is the plant cell. Interphase stage, DNA will multiply, all the other material will multiply, then nucleus will divide and move into the two poles, then the cytoplasm will divide. Right, so this is the chart in table. So in your book there's a slight mistake in the table, please correct it. Right, so this is the interphase. Interphase is divided into 3 stages: G1, S, and G2. Then mitotic phase, cell nuclear division phase, then cytoplasm division phase, cell division C. Right, interphase, mitosis phase, cell division stage. During interphase or G1, it is first stage is G1. During that we call primary growth phase of the cell. Stage synthesis. It means DNA will replicate. Then G2 is secondary growth phase. Then the cell will enter the nuclear division stage with 4 phases. After that, cytoplasm will divide. Those are the different phases of the cell division, right? This is called interphase. First growth phase, DNA replication stage, S phase, G2. Growth phase, then the cell will enter nuclear division stage, prophase, metaphase, anaphase, telophase. Then cell will enter the third phase, cytokinesis. Then cell can differentiate and do another function. Or else the cell can move to the growth stage and it will move as cyclic process. It will move as a cyclic process. So large number of cells will be produced. Oil cells can modify and perform different functions. Like if it from a root hair, it will adapt to absorb water. Then DNA is the deoxyribonucleic acid. This is responsible for the transfer of hereditary material from mother to the daughter cells. So every body has same amount of DNA. So DNA molecule is packed into a chromosome. This is called chromosome and this is called gene. It is bind together with the centromere, and these are DNA molecules are inside the chromosomes. And if usually in our body cells, like in our hand, in our face, in our hairs, we have diploid number, 2 copies of chromosomes, as you can see here, red and blue, 2 copies. But when we produce our reproductive cells, like ovules by females and sperms by males, this 2n will become n. Then we call haploid. Usually our body has 2n cells, 2 set of pairs, 2 copies of each chromosome. When it move to the reproduction, we will produce n number. So when another sperm mix with an ovule, it will become 2n again, haploid and diploid, right? Here you can see parent body, like our hairs, face, hands, legs, 2n. During mitosis, they will produce large number of similar 2n cells. But when they produce germ cells, sperms and eggs, the number of genetic material become half, n. So when the egg combined with the sperm, it will produce 2n again. So you can maintain the 2n nature throughout the life cycle. Because during reproduction only it is halved. During meiosis, mitosis, genetic level is maintained as diploid. So cell division is of 2 types: mitosis, anunana vibhajanaya, and meiosis, unana vibhajanaya. If you compare it, mitosis has only 4 stages. Meiosis, 8 stages. Mitosis happened in somatic cells, means cells of the body, in face, in a stem, plant, leaf elongation, production of flowers, fruit enlargement, somatic cells. Meiosis happened in germ cells. Mitosis purpose is cellular proliferation, producing large number of cells. But here, meiosis, sexual reproduction. Here produce 2 diploid daughter cells, but this one produce 4 N or haploid daughter cells. The chromosome number remains same, but here chromosome number is half than the original daughter cell. Genetic variation, no. But here meiosis, genetic variation present. In both mitosis and meiosis, the plant will produce new cells. Right, now let's look at the mitosis and meiosis. Mitosis, we have parent cell, certain amount of DNA material, it will duplicate, it will double. And one set will go to the one daughter cell, another set will go to another daughter cell, second daughter cell. So 2n number will be maintained here. But during, this is anunana vibhajane, but during unana vibhajane, parent genetic materials will double, then halves will go to the 2 daughter cell, another will go to another 4 daughter cells. So ultimately 2n, this will be n. Right, let's look at the anunana vibhajane or mitosis. So nucleus divides into 2 daughter nuclei. So all the daughter nuclei will contain same, identical or 2n genetic material. After nuclear division, cell will divide. Here, this is the mother cell, DNA present, they will duplicate, replicate, right? So here you can see, then half will go to the one cell, the other half will go to the other part. So here you can see, if you look at the genetic number, material, it is same. So 2n is maintained. So this happened over 4 phases: Prag Kalava, Yoga Kalava, Viyoga Kalava, Anta Kalava. First is called prophase. So before that, I told you the cell will undergo interphase. So chromosome doubling is happening during interphase. Then the cell will go to the prophase. So during the prophase, chromosomes become thickened and it will appear. Spindle fibers also appear. Then metaphase, all the chromosomes will line up in spindle. Tarkwe samaketa chromosome පැමිණෙනවා. Chromosome are line up in the equator of the spindle. Then this part can move to the one pole, the other set can move to the other pole. It can separate like this and move to the two poles, right? Anaphase, this paired chromosomes separate and move to the opposite poles. Then telophase, chromatids arrived at the poles and they form a nuclear membrane. Then the cell cytoplasm will divide and it will form two cells. First nuclear division, it is completed. Now then cell will divide and produce two cells. Then the cell will enter the cell division stage, cytoplasm division stage, that is cytokinesis, and it can continue this. So mother cells and daughter cells are equal in chromosome number and features. Then let's look at meiosis, that is Unana Vibhajana. During that, nucleus divides to form two daughter nuclei. And remember, daughter nuclei containing not 2n, but half number of chromosomes of the original nucleus. During meiosis, first division, daughter nuclei will give only half number of chromosomes to the daughter nuclei, right? From mother to daughter nuclei contain half number of the chromosome. So this will happen during gamete and spore formation. During ovule and sperm formation, this will happen. I know if you don't have initial knowledge about meiosis and mitosis, it's very difficult to understand, I know, but can't help because A-level, this knowledge is important. This is, once again, A-level things, right? So if you have not followed this thing, it may be difficult to understand. So it has two phases, meiosis 1 and meiosis 2. Meiosis 1, single cell divided into two. So two cells will form. Then these two cells divide again and ultimately will produce four cells, right? So meiosis 1, it has prophase 1. Prophase 1, there's a special thing. It is very much similar to the previous mitosis, right? But there's a specific thing is going to happen. We call crossover. Avatarane sidu වෙනවා meiosis stage 1 prophase 1. අවතරණය සිදු වෙන්නේ ඌනන විභාජනයේ ප්‍රාක් කලාව එක අවධියේදී අවතරණය සිදු වෙන්න පුළුවන්. So it is happening in meiosis stage 1, prophase 1. What will happen when the similar chromosome, homologous chromosome pair up? These chromosomes can join from different places. We call chiasmata. From these places, chromosomes can join together and they separate. Fragment of DNA from mother and father can exchange. So ultimately, genetic material can be exchanged. It is explained here. This is the synapses. So pairing of homologous chromosome. So when they pair, sometimes these parts can overlap like this. So when they separate, red color can be fixed to orange color part can fix to red color, right? So material can be exchanged, crossover. You called it crossover in single avatarane. So there could be several such places. As you can see here, several such places. So most of the genetic material can be mixed between parents. It leads to the genetic diversity or genetic variation. Then the cell will enter metaphase 1, that is cells genetic material aligned along the equatorial plate. Then they will move to the poles and complete the first stage, right? Here 2n during metaphase, they arrange in this equatorial plate. Then the chromosome will move to the poles and form two nuclei. So those are n. Mother cell is 2n, but these are n. Then this will undergo second division and this will produce another two cells. So process is similar. Prophase 2, metaphase 2, anaphase 2, telophase 2, similar thing. So these two cells can undergo cell division again and ultimately will produce four daughter cell after kinosis. So from 2n mother cell, 4n cells can be formed after meiosis. This is explained. If you cannot understand or difficult to understand, the best thing is in the YouTube, you have several videos, colorful, and it has animations. So you can understand the process. You can use these color pictures as well. Right. So finally, what are the importance of mitosis and meiosis? What is the importance of Anunana Vibhajane or mitosis? It is helpful in asexual reproduction. අලිංගික ප්‍රජනනයේදී වැදගත් වෙනවා. Why? During cell growth and cell replacement. Cell growth and cell replacement. When a cell replaced with another cell, mitosis happens. Then this help to give genetic stability because all the cells will be genetically identical. Therefore, that's the reason for using cuttings to propagate tea, rubber. Why? When we use cuttings, mother plant and daughter plant's characters will be exactly similar. Significance of meiosis. ඌරණ විභාජනයේ වැදගත්කම. It is important in sexual reproduction. Sexual reproduction involves two parents, mother and father, and produce offspring. So the mother and father's genetic material will be combined. So daughter cells have unique genetic characters. හැදෙන සෛලවල ජාන සංයුතිය අම්මගේත් නෙවෙයි තාත්තගේත් නෙවෙයි දෙකේම combination එකක්. And it is important to maintain genetic variation because if the genetic variation is genetic variation is zero, what will happen? All the plants will be very much similar. All the plants will be susceptible for certain diseases. Yield will be very high. All the plants will give yield at the correct same time. But if there's something pest and disease issue, entire population can wiped out. So if you want to introduce genetic variation into the population, we have to promote meiosis. So in meiosis, genetic variation is maintained. How? Through crossover and through independent assortment. We discussed crossover. Independent assortment means during anaphase, I hope you can remember, chromosomes of each bivalent pair separate randomly, independently from each other. As an example, one came from mother, the other one came father. And there are 4 possibilities for different combination. Only blue can combine and give this type of combination. Red can combine and give this sort of combination. If not, blue and red can mix and give this sort of combination. So this is because genetic material can independently move. We call it independent assortment. ස්වාධීන සංරචනය. ක්‍රෝමසෝම් වලට පුළුවන් වෙන් වෙලා Tamanට කැමති එක්කෙනෙක් එක්ක ස්වාධීනව සම්බන්ධ වෙන්න. තවත් එක්කෙනෙක් එක්ක. එතකොට ඒකට අපි කියනවා ස්වාධීන සංරචනය කියලා. This lead to the genetic variation. Right. Okay. With that, let me finish the theory part. This is the assignment. Explain Anunana Vibhajane or mitosis. You can refer the book and explain. Then compare mitosis and meiosis. Just now we discussed. And you can submit the answer when you come to the class exam. Right? You cannot download pictures and paste. You have to write down your assignment, right? No images, no pictures from internet. You can read the book and write down and submit to a assignment on 23rd February when you come to the exam. So you can score about 30% marks, right? If you delay, you will face consequences. Please don't delay your submission, right? Then for the next week, we are going to have practical class. When you attend the practical class and learn, for that process, I will give you 10% mark. So when you come to the practical class, please bring A4 sheets and drawing sheets, few sheets like 6. Then you need the file cover to keep this and to draw pencil, eraser, ruler, file cover, probably a blade. So you have to cut certain things and observe, right? So these are the material required for the class, right? When you come to the class, please remember to bring this material. Right, with that, let me wind up my session. long day, and I will share my lecture notes with you tonight. So I hope to meet you all on next Saturday. Please, if you want to change the day, please let me know. Otherwise, it will be difficult for me to conduct the practical class, because I'm going to group you, all the students in this Saturday group, into 4 groups, and I'm going to rotate it into different practicals, right? So I have to maintain certain student number, otherwise it will be difficult to conduct. So please, if you are not coming, please let me know in advance, otherwise you will face consequences, right? Because it will be difficult for us to manage large student number for practical, right? Only there will be very few practical classes. When crop physiology, you have completed the practical class. In my class also, there will be a simple practical class. They will be directly related to the practical examination, right? Okay. With that, let me finish. Have a nice weekend. I will not have a nice weekend because I have another class on Sunday. You can enjoy the Sunday, right? Okay. Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you, sir.
See you on next week, Saturday.
Okay, thank you, sir.